avoid using avoid using these uh, the chemicals, the chemical symbols. So it's the same thing that applies to the hydrochloric acid and the sodium hydrogen carbonate. So whenever we are writing them, we are not supposed to indicate their symbols or their chemical formula. I mean, the mixture changes to violet or the blue color. Is it a must to indicate the volumes in the burette tests? That is a question a, from Ruth. Not a must, unaweza tumia approximate volumes because even if we indicate it this way, as long as the order has been followed, you start with sodium hydroxide, then followed by the copper sulfate. Is copper sulfate added in dropwise? Mildred, yes. Copper sulfate is normally added dropwise. Then Winnie is asking, can you say add the sodium hydroxide and solution until it clears? Winnie, what does that word clears mean? You can use your you can respond it by audio. Can you say the word? What do you mean by the word clears until it clears? Winnie, can you clarify that?
I mean it turns colorless.
Remember, you start with the sodium hydroxide, then the copper 2 sulfate. So the mixture, you do not have to indicate it until it clears. You just say add sodium hydroxide, then followed by copper 2 sulfate dropwise. So that is the correct way we are supposed to indicate that. Then the observation is as given there. The mixture changes to violet, or the blue color of the copper sulfate is retained if the proteins are absent. But if they are present, that is the expected color there. Mr. Paul, you can scroll up so that we can move to the next part. The next part there should be the grease spot tests or the emulsion tests, or those are the terms that we normally use. Yes, there is a question. First, I want to respond to that before we continue. I think I've responded to Winnie. In burette test, do we add equal amounts of sodium hydroxide? Yes, you can say equal amount, or you can be specific on the amount of food substance that you will be able to add. But the word equal is normally, or in most cases, we use it under the reducing sugars. Here we only indicate add 2 cubic centimeters. But if you also use this word equal amount, if you had indicated to the food substance add equal amount of sodium hydroxide, it will also be correct for that part. Mr. Paul, if you can get me, you take us to the next part. The next part should be on the grease spot tests. Not that way. They still on the table. Yeah, they are. The grease spot tests or the emulsion tests, all tests for the lipids. So grease spot tests, emulsion tests, all the tests for lipids, if it is the food substance that we are investigating. In this case, on the procedure, We indicate, smear, or apply the full substance on a filter paper, then dry it over a flame and then observe it against light. That is if you are using the grease spot test. Normally, students are advised that whenever they are drying that filter paper, they should not put it directly to the flame. They just put it closer, but without igniting that piece of paper. Then after it has dried, they can observe it against light. The observation that is required is that a permanent translucent spot will be formed, which indicates that the repeats are present. So when we smear or apply the full substance on a filter paper, then you dry it over a flame. If a permanent translucent spot is formed, that is an indicator that the reducing sugars are, no, the repeats present, sorry. You can also use these emulsion tests that we normally use the ethanol. So you say to the full substance, add 3 drops of the ethanol. So the ethanol here is added dropwise, and whatever is formed is that a white emulsion will be formed. And when a white emulsion has been formed, that is an indicator that the repeats are present. There is someone who is requesting as that is Winnie, going back to the burette test on the screen. So we'll be able to do that. That is whatever we are having there, Winnie. So test for proteins, we refer to that as the burette test. Yes, Sylvia, in burette test, can color change to purple? Yes, but we normally indicate violet. The purple and violet are the ones that we are expected. I think light purple, it should be violet. That is the correct color change that we are supposed to give. Purple, even when we are doing that experiment, we do not observe the purple color. Instead, tunaona the violet within that part. I hope, Winnie, you have observed within that burette test, that is whatever we were supposed to have. But if there is any question still, you can be able to ask. I've responded to Sylvia on the change of color to purple, and I've said the correct one should be violet. We can continue. On the next part, it's on the DCPAP tests. DCPAP in full is dichlorophenol indophenol. It's not a must for you to indicate that full name. The abbreviation is acceptable. Mr. Paul, umeenda beyond. Tuko slightly, there. So DCPAP tests all tests for the vitamin C or the ascorbic acid. This is a unique test because in all the other experiments, we start by placing the food substance, then the chemical substance to that food substance. But for this one, we normally add the DCPAP first to the test tube, then you add the food substance dropwise. So DCPAP first into the test tube, then followed by the food substance, and it should be added dropwise. What I've not mentioned is that the DCPAP tests can also, or when it has been indicated the test, you can say test for vitamin C. C should be capital. When we are instead of vitamin C, you can also say ascorbic acid. So the procedure is as I've explained there. Then when you are indicating the observation, you will indicate the DCPAP is decolorized. the DCPIP is normally blue in color. So if a student says the DCPIP changes from blue to colorless, still it will be okay. The color of DCPIP changes from blue to colorless will be correct. And you will say there vitamin C present. But if the DCPIP is not decolorized, then it means that the vitamin C is absent. So that is on that vitamin C where you use the DCPIP to test for that vitamin C. And I've said this DCPIP should be indicated in capital letters, no dots between those letters. It's not a must. If you can manage to remember that word dichlorophenol, indophenol, it will be okay. But if you will not be able to remember that, just indicate it the way we have done it there. The next part is test for the non-reducing sugars. So for the non-reducing sugars, what you are supposed to do under the procedure. So for the non-reducing sugars, sorry if I can emphasize this, normally ukiambiwa uandike the test, unaandika test for non-reducing sugars. Chemical test, we normally use number of chemicals. So they normally guide you. If they will test you on the non-reducing sugars, I mean, within that column up there, they will be able to give you an appropriate word that will enable you to use that. SC is asking a question, I can respond to that. In the test for reducing sugars, when the color stops at yellow, can you say few amounts of reducing sugars present? True. In fact, I should have mentioned that the quantities under the reducing sugars can be indicated. Like now, if the color of Benedict solution changes to green, a student should indicate little amounts of reducing sugars present. If it is changing to yellow, moderate amount. If it is changing to orange or brown, high amounts of reducing sugars present. So the amounts can be indicated. It's correct, especially if you do the boiling and the color persists at either green or it persists at a yellow or it persists at brown. So you can be able to indicate the amounts that way. But if you indicate reducing sugars present again, you will not be penalized. It will be correct. Whether they are low, moderately high, or very high, still it will be okay. So I was on the reducing sugars, and I was saying this, that on the reducing sugars, on the procedure part, you indicate that to a food substance, you add hydrochloric acid, then a solution of sodium hydrogen carbonate. Mr. Paul, you can move us so that we can go to get clear here that way. Thank you. To a food substance, you add hydrochloric acid, then a solution of sodium carbonate until no effervescence is observed. So first, the food substance is placed in a test tube, then hydrochloric acid. The amounts should not be specified. It's not a must. And the sodium hydrogen carbonate, it's normally added little by little until there is no effervescence that is being observed. After that, you are supposed to add equal amount of Benedict solution. Here, we are supposed to get this, that equal amount of Benedict solution to the mixture that we would have formed. Then after that, we heat the mixture. Again, because we are using this word Benedict solution, B should be capital, the apostrophe should be indicated as shown within that part. When we are indicating the observation, We only measure our observation on the Benedict solution. So we should indicate that the Benedict solution changes from blue to green to yellow to orange to red. That order of those colors for the Benedict solution should be followed. So that is what is expected on the observation. But if the non-reducing sugars were not present, then it means that when you are adding the Benedict solution, those color changes, all the blue color of the Benedict solution is going to be retained. There are other questions related to this table that I should be able to mention here. You can be asked the reasons for applying some processes during the experiment. Like now on this part of the reducing sugars, you can be asked, what is the role of this hydrochloric acid? Why should we add the sodium carbonate? And lastly, why should we add the Benedict solution? Like for the first one, I really want you to participate and respond to this one. I can pick someone randomly to respond to that. Amani, why do we add hydrochloric acid to this food substance suspected to be containing the non-reducing sugars? Amani, respond to that if you can get me. Amani, are you there? Brian Koech, kindly respond to that question. Why hydrochloric acid to the food substance containing or suspected to be non-reducing sugars? Brian Koech, someone to volunteer herself or himself and respond to us that. Someone to volunteer.
To hydrolyze non-reducing sugar to reducing sugar to non-reducing sugar.
Nasema the other way around.
Non-reducing sugars to reducing sugars. That is good. That is good. That is exactly why we add the hydrochloric acid. What about addition of sodium hydrogen carbonate? Someone else to respond to that. Why should we add the sodium hydrogen carbonate until there is no effervescence? Levi?
So as to neutralize the excess hydrochloric acid.
Good. To neutralize the excess hydrochloric acid. Then after the non-reducing sugars have been converted to reducing sugars, that is now when we use the Benedict solution so that we can be able to check whether the reducing sugars are present or not. Now, you can get questions on deficiency diseases related to the food substances that we have discussed there. Like now I'm starting with the vitamin C. Which deficiency disease will someone get due to the lack of that vitamin C? A volunteer also, vitamin C. If someone lacks vitamin C, which deficiency disease will that person be suffering from? Yeah, someone answer. Yes, Lucy, you can respond.
Scurvy.
Good, scurvy. Good. That is the disease that that person will be suffering from. What about if it is proteins? Proteins. Which deficiency disease will someone suffer from if within the body of that person atakuwa hana nini, hakuna Onchonga? Yes, Onchonga, I've given you the
Protein.
opportunity.
Lack of proteins leads to kwashiorkor.
Kwashiorkor. That is correct, Onchonga. And take note of this. That question, most teachers prefer or like testing it because students normally misspell the word kwashiorkor. So check on the correct spelling for that word. And whenever you are given that, you should be able to respond to that question correctly. Something related to this. A student can also be given an application question whereby either a sample of urine is taken from an individual, then it will be used to test for the presence of the reducing sugars within the urine. And maybe you may be asked to describe the procedure on how you are going to investigate the presence of the reducing sugars within the urine. So it's the same procedure that we have given there. You place the urine sample in a test tube. Then to that urine sample, you add equal amount of Benedict solution. B should be capital with an apostrophe there at the end. That is between T and S. Then you heat to boil the mixture. Then you make the observation. If the mixture changes from blue to green to yellow, it indicates the presence of the reducing sugars within the urine. And for that case, I'm asking a question related to that. In case someone will be having glucose within the urine, which disease will that person be suffering from? It's an application question. Let me hear from you. Someone to volunteer again. What leads or which disease leads to the presence of glucose or reducing sugars within one's urine? Faith, you can respond.
Diabetes mellitus.
Good. The diabetes emeritus. That is the disease that individual will be suffering from. So, that is a brief summary of what we normally get on this part of the first part, which is on the food test. The next area that I want us to look at is on the classification and the dichotomous key. Mr. Paul, you can scroll up. One thing that we should understand on this classification is that we are supposed to know this, that whenever we are writing the taxonomic names, they should always start with a capital letter. The word plantae, you will be given maybe a bean seed. Lucy, Anna, Uriza, we pardon on the type of diabetes. It is diabetes mellitus. Diabetes mellitus, the type of diabetes that one will be suffering from if there is presence of glucose or reducing sugars within the urine. So I was saying this, that on classification, always ensure that you write the taxonomic names in capital letters. Like now you are given a bean seedling, then you are asked to classify that seedling. Of course, the class to which this seedling belongs to is the Dicotyledonea. But if it is the kingdom that it belongs to, it should be kingdom Plantae. So Plantae is the one that should be in capital. So take note of this. When I'm saying it should start with the capital letter, some may indicate capital K, but the other part is in it is on that part, on the actual name of that group of organsim, plantae, P capital, or dicotyledonine. So that is one thing that we are supposed to consider there. When we are giving those groups to which the organsims belong, we should use the observable features to place those organsims in their specific classes or in their specific groups. Like now you are given a bird, like now an eagle, you can classify it under the class, not the class, but yeah, within the class Fs, A capital Fs. And the reasons why we will be able to give it that way, it is because it has got feathers, its mouth is modified to form the beak, among other reasons. So the Fs have indicated it there under the class that. So we should always use those the characteristics that we can be able to observe within that organsims. Still on the classifications, we have got this part that is very common also that we refer to as the dichotomous key. The dichotomous key is a part of the classification, and on this dichotomous key, we are supposed to understand the following when we are constructing it, that these are the formats that we are supposed to know. You can be given an organsim that you will use to construct the key. The organsims are given, the characteristics you are supposed to select on your own. Then you construct a dichotomous key. Another format is that you are given organsims and key to give the steps for identification of the organsims. So you are given the dichotomous key, and the statements. Then you are supposed to respond on that. Then we are given organsims and constructed key with gaps to be filled. So you can be given organsims and a key that has been constructed but not complete, so that you can fill the gaps within that part. So those are all the common types of dichotomous key that we normally get. The common features that are used in the dichotomous key within the plants. But before I go to that, there is someone asking a question here, Mildred. High amounts diabetes mellitus, low amounts diabetes insipidus. One thing that we are supposed to get is this, that this diabetes, in fact, we are not supposed to have glucose within our urine. And if it is there within our urine, it means that this glucose is not reabsorbed into the body and it's not broken down, or it's not converted into the forms that it should be within the body, meaning that there is a defect with the secretion of the insulin. So the main one that we are supposed to have given for our case there is the diabetes mellitus. That is what we were supposed to have given within that part. I can continue. So you are on this part of the common features used in the dichotomous key. For plants, we use the leaves. Mainly the leaves are the ones that we use, but also we can use the stems. We can also use the fruits. We can also use the flowers. If we are using the leaves, we can look at the type of leaf. We have got two types of leaf. We have got the simple leaf. We have got the compound leaf. The simple leaf is a single leaf. Simple leaf is a single leaf. Just one leaf. That is what we refer to as a simple leaf. Sometimes students do not get what it means by this part of a simple leaf. So it's a single, it's only one. But for the compound leaf, it's a leaf that has many leaflets within it. So iko na one main stalk connected to leaflets connected to that main leaf stalk. So when that is the case, then we refer to that type of leaf as a compound leaf. The compound leaf can be in different formats. It can be a trifoliate if it has got 3 leaflets within it. It can be a palmate if it is having 5 leaflets within it, also referred to as the digitates, palmate or digitates. It can be pinnate if it is having pairs of leaflets arranged within the main leaf stalk. It can be bipinnate if it is a leaf that has got a main stalk and that main stalk is branched. That is what we refer to as bipinnate. You can take us to the next part, or you scroll up, Mr. Paul. So those are the ones that we normally get within the compound leaf. That is on the type of leaf under that part of the compound leaf. You can also use this part. Yeah, thank you. On the margin can be smooth or entire, or the margin can be serrated or dented. There is a slight difference between serrated and dented. Remember, this part that we are using, the dented, it's not smooth. It means that the zigzag margin. But if it is very sharp, that is when we refer to it as dented. If those it's wavy like, that is when we refer to it as being serrated. I can illustrate it for these. For that is for serrated. For dented, it's normally very sharp. So that is whatever we are supposed to get on the difference between those two. Then we have got the veins. Of course, the veins are very common. It can either be a network or parallel. There is a question I can respond. Could you please explain the palmate leaf again, but in simpler terms? A palmate means that it has got 5 digits, just like a palm. So if those leaflets are like that, 5 of them that way, then we refer to this one as palmate. I've tried to illustrate it that way. Hii ingine yenye wanasema trifoliate, kama iko this way, then nakuwa na hiyo your leaf that way, you can see there are 3. We refer to it as a trifoliate. Then we have got the other one hiyo yenye tumeita the the pinnate, tunakuwa this way. that is within the main stalk, tunakuwa na 2 leaflets within that main stalk. Bipinnate, I can try to show it that way. So within this one is the main stalk, then there is a branch, then unakuwa na leaflets within these sides. So if that is the case, then we refer to that as the bipinnate. So I think the one who was asking for the clarification of that, or of those terms, have illustrated them there. For the venation, I've already emphasized on that. Kuna hii ingine yenye huwa tunasema the apex being pointed or rounded or lobed. Lobed is something that looks this way. If you look at the apex, in some leaves, unapata iko this way. So hii part, hiyo ndio huwa tunaita the rope. Some unapata iko bi-lobed, ziko mbili. Oh, sorry. I can be able to show it in a better way. So if it appears like that, then it's bi-lobed. So these structures are the ones that we refer to as the lobe. So it can be bi-lobed, it can be lobed that way. Then we can also look at the way it's normally attached to the stem. Some of the leaves use the petiole to attach itself to the main stem. Like now the kales and the beans. But like now the monocots, they normally use a sheath to attach themselves to the main stem or to the stem. So that is something that we're supposed to know when we are in constructing our dichotomous key. Then we have got the texture. On the texture, we can be able to feel the leaf, and we can be able to classify or to identify it as being smooth or being rough. So we can use either the smooth or the rough just on that part of the texture. Like now you can use this one, Tradescantia. Its ramina or its surface is normally smooth. If you use the Rantana Camara, it's normally rough. So that is on the texture. We can also use the thickness of the leaf. Some of the leaves are very thick, others are very thin. So you are given like now the leaf of the Tradescantia again, it's normally thick or succulent, and the other one is normally thin. So you can also use that as a structure to differentiate the different plants. You can also use the color of the plant. We have got the green, we have got the purple, we have got the other one that we refer to as the variegated. Those plants that have got the chlorophyll